Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with pyruvate dehydrogenase complex. In today's session, we will start with regulation of enzymes in glycolysis. Here we will see allosteric mechanisms and covariant modification of pyruvate dehydrogenase. Then we have importance of pyruvate dehydrogenase, in which we will see that this reaction is completely irreversible process. Then there is clinical application of glycolytic enzymes, in which we will see lactic acidosis, which is seen in hypoxia, shock, pulmonary failure, alcohol abuse, and diabetes mellitus. Good morning, everyone. Let us start the topic. Regulation. PDH or pyruvate dehydrogenase is subject to regulation by allosteric mechanisms and covariant modification. Allosteric inhibitors are the products NADH and acetyl-CoA. PDH is covariantly modified by phosphorylation by PDH kinase and dephosphorylated by PDH phosphatase. The dephosphorylated form of the enzyme is active form. Hence, activators of PDH kinase like ATP, NADH, and acetyl-CoA inhibit PDH reaction. The PDH phosphatase is activated by calcium, magnesium, and AMP. This will increase the rate of PDH reaction, whereas PDH kinase is inhibited by Ca plus or calcium. When there is adequate ATP and acetyl-CoA, the enzyme is inhibited. Coming to importance of PDH or importance of pyruvate dehydrogenase, it is completely irreversible process. There is no pathway available in the body to circumvent this step. Covariant glucose through this step is converted to acetyl-CoA, from which fatty acids can be synthesized. But the backward reaction is not possible, and so there is no net synthesis of glucose from fat. Pyruvate may be channeled back to glucose through gluconeogenesis, but when pyruvate becomes acetyl-CoA, it cannot go back. Thus, pyruvate dehydrogenase step is the committed step towards oxidation of glucose. Energetics. The NADH generated in this reaction enters the electron transport chain to produce 2.5 ATP molecules. Pyruvate dehydrogenase is regulated by end product inhibition as well as by covalent modification. Phosphorylation of the enzyme by a kinase decreases the activity of the enzyme. Coming to clinical applications of glycolytic enzymes. Lactic acidosis may be seen in hypoxia, shock, pulmonary failure, alcohol abuse, and diabetes mellitus. Deficiency of glycolytic enzymes. These conditions are rare, out of which pyruvate kinase deficiency and hexokinase deficiency are comparatively common. Though rare, these deficiency states can lead to hemolytic anemia because energy depleted RBCs are destroyed. Hexokinase deficient RBCs have low level of 2,3-BPG and a high affinity for oxygen. On the other hand, deficiency of pyruvate kinase leads to decreased oxygen affinity and since the 2,3-BPG levels are high. Inherited altolase deficiency causes hemolysis. In PFK deficiency, muscle weakness is seen. Pyruvate dehydrogenase requires thymine pyrophosphate, that is TPP. This explains the serious afflictions in beriberi due to thymine deficiency. Thymine pyrophosphate deficiency in alcoholism causes pyruvate accumulation in tissues. Inherited PDH deficiency may lead to lactic acidosis. Coming to pyruvate as an junction point. Pyruvate occupies an important junction between various metabolic pathways. It may be decarboxylated to acetyl-CoA which enters the TCA cycle or may be utilized for fatty acid synthesis. On the other hand, pyruvate may be carboxylated to oxaloacetate which is used for gluconeogenesis. In this diagram, we can see that pyruvate as a junction point. Pyruvate can be converted to lactate in the presence of enzyme lactate dehydrogenase, or it can be converted to alanine in the presence of alanine transaminase, or it can be converted to acetyl-CoA by the enzyme pyruvate dehydrogenase, or it can be converted to oxaloacetate by enzyme pyruvate carboxykinase, or it can be converted to phosphoenol pyruvate through oxaloacetate with the presence of enzyme phosphoenol pyruvate carboxykinase. Coming to the next point, gluconeogenesis. Definition: it is a process by which glucose molecules are produced from non-carbohydrate precursors. These include lactate, gluconeogenic amino acids, glycerol part of fat, propionyl CoA derived from odd chain fatty acids. Coming to the site of gluconeogenesis. Gluconeogenesis occurs mainly in the liver and to a lesser extent in the renal cortex. The pathway is partly mitochondrial and partly cytoplasmic. Key gluconeogenic enzymes are pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. In this diagram, we can see glucogenic pathway which starts from the bottom. In glycolysis earlier, glucose 6-phosphate was formed by glucose in the presence of enzyme hexokinase, but now in glucogenic pathway it converts the opposite reaction with the help of enzyme glucose 6-phosphatase. In glycolysis, glucose 1,6-bisphosphate was formed by glucose 6-phosphate with the presence of enzyme phosphofructokinase. Now in glucogenic pathway, it forms the opposite reaction via fructose 1,6-bisphosphatase. Earlier, it used to form pyruvate by phosphoenol pyruvate by pyruvate kinase, and later this pyruvate used to convert into oxaloacetate in the presence of enzyme pyruvate carboxylase. Now in gluconeogenic pathway, the opposite reaction can take place via enzyme phosphoenol pyruvate carboxykinase. Coming to key gluconeogenic enzymes. Gluconeogenesis involves several enzymes of glycolysis, but it is not a reversal of glycolysis. The irreversible steps in glycolysis are circumvented by 4 enzymes which are designated as the key enzymes of gluconeogenesis. In this table, we can see the key enzymes for glycolysis and versus gluconeogenic enzymes. Earlier, there was pyruvate kinase at step 3 in glycolysis, but in gluconeogenic enzymes, we have pyruvate carboxylase and phosphoenol pyruvate carboxykinase. In glycolysis, phosphofructokinase was present at step 3, but now gluconeogenic enzymes, it has fructose 1,6-bisphosphatase. Earlier in glycolysis, it used to have hexokinase at step 1, but now in gluconeogenic enzymes, you have glucose 6-phosphatase. Coming to key enzymes, pyruvate carboxylase reaction. Pyruvate in the cytoplasm enters mitochondria. Then carboxylation of pyruvate to oxaloacetate is catalyzed by mitochondrial enzyme pyruvate carboxylase. It needs coenzymes myotin and ATP. In this diagram, we can see first step in gluconeogenesis. Pyruvate is converted to oxaloacetate with the help of enzyme pyruvate carboxylase, and ATP and biotin together are the coenzymes. Malate aspartate shuttle. The carboxylation of pyruvate, that is the previous reaction, takes place in mitochondria. So oxaloacetate is generated inside the mitochondria. This oxaloacetate has to be transported from mitochondria to cytosol because further reactions of gluconeogenesis are taking place in cytosol. This is achieved by the malate aspartate shuttle. First, oxaloacetate is converted to malate, which traverses the membrane and reaches cytoplasm. Malate is then reconverted into oxaloacetate. Malate dehydrogenase is present in mitochondria and cytoplasm. Oxaloacetate may also be transported as aspartate formed by transamination of oxaloacetate. When alanine is the substrate for gluconeogenesis, malate shuttle predominantly operates because NADH is also required in cytoplasm for the glucogenesis to continue. On the other hand, when lactate is the substrate for gluconeogenesis, the aspartate shuttle operates because sufficient NADH is available in the cytoplasm by the LDH reaction. In the cytoplasm, PEPCK, that is phosphoenol pyruvate carboxykinase enzyme, then converts oxaloacetate to phosphoenol pyruvate by removing one molecule of CO2. GTP or ITP donates the phosphate. The net effect of these two reactions is the conversion of pyruvate to phosphoenol pyruvate. This circumvents the irreversible step in glycolysis catalyzed by pyruvate kinase. In this diagram, we can see PEPCK or phosphoenol pyruvate carboxykinase. Oxaloacetate is converted to phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxykinase and removal of one molecule of CO2, and GTP is converted to GDP. Coming to the next point, partial reversal of glycolysis. The phosphoenol pyruvate undergoes further reactions catalyzed by glycolytic enzymes to form fructose 1,6-bisphosphate. All these reactions are freely reversible. Fructose 1,6-bisphosphate is then acted upon by fructose 1,6-bisphosphatase to form fructose 6-phosphate. This will bypass the step of phosphofructokinase reaction. Just as seen in the reaction, fructose 1,6-bisphosphate is converted into fructose 6-phosphate plus Pi in the presence of enzyme fructose 1,6-bisphosphatase. Then fructose 6-phosphate is isomerized to glucose 6-phosphate by the freely reversible reaction catalyzed by hexose phosphate isomerase. Next is glucose 6-phosphatase reaction. The glucose 6-phosphate is hydrolyzed to free glucose by glucose 6-phosphatase. As in the reaction, glucose 6-phosphate plus H2O gives glucose plus Pi. Glucose 6-phosphatase is active in the liver. It is present in the kidney and intestinal mucosa to a lesser extent, but it is absent in the muscle. Here ends our session. Kindly like, share and subscribe the channel. Thank you.